Good morning, good afternoon, everyone. So as usual, this meeting will be recorded, and so we plan to post this on YouTube. See if something occurred during the meeting you want us to crop out, please reach out before the end of the day. Today we have two items. Sarah Rudiger's groups will present, so Alicia or Yuyan Wang will present some analysis on standard oddball. And then I think on SLAP2 data, maybe she's touching over modalities. And then Carter will introduce a number of notebooks that he has been working in the last few weeks to sort of help out the data release publication. And so we'll speak to that in the second half. So we need to keep at least 20 minutes for the end. Okay. That being said, Alicia, welcome. Maybe introduce yourself and start the meeting.
Right. Okay. Hi, everyone. Can you see my screen now?
Yes.
Yeah. Okay. Hi, everyone. My name is Yuyan Huang, and I'm really happy to share my final year BSc project analysis of the OpenScope SLAP2 dataset, which asks whether the synaptic inputs in the mouse V1 adapt during a standard mismatch paradigm. And I'm supervised by the Dr. Sarah Rudiger. Yeah. And so to start with, I will start with a little bit background. And so in the visual oddball paradigms, the larger responses to the oddball may often be interpreted as the evidence of mismatch signaling, but they may not only limited to that. So they may also shaped by the interacting mechanism, including the complex temporal sequence structure, the local adaptive gain mechanism, and the recurrent cortical processings and others. And most of the previous work has examined at the level of a neuronal output like the spiking or the somatic calcium or the population activities. So the SLAP2 data allows us to ask from a quite different perspective from the dendritic perspective. So my main question is, how do the synaptic inputs into V1 adapt during a visual mismatch paradigm? Yeah. So I mostly use these 5 mice and 5 experiments for the analysis. But to note that there are 2 data, they lack the block 2 due to the improvement of the experiment design. Yeah.
Just a quick question, Alethea. Are you changing slides or not? Because I'm still seeing the title slide. I just want to make sure we're on the same page. Okay.
Oh yeah, I'm actually changing the slides. Sorry.
Okay. I only saw the title slide.
Okay.
That's good.
So yeah, I will not like play the presentation mode if it doesn't change. I will just like list as this is fine, right? Okay. Yeah. So basically the second slide is only the background, and the data slides is to say that I use these 5 mice, but like 5 experiment, and 2 of them like may lack the block 2 due to the improvement of the experiment design. Yeah. And so to begin with, I have some like response matrix that I calculated. So this is a example of the standard in oddball block 1 in the standard stimulus, and one second trial of the ROI 0, and I measured the absolute response, which is the main DFF during the stimulus window from 0 to 0.343 seconds, and the pre-stimulus, which is the baseline response, from the minus 0.343 to 0 seconds, and the baseline subtracted, which I also called the relative response later, which used the stimulus absolute mean response to minus the baseline mean response. And the reason why I add this relative response is because when I when I visualized the same ROI but under different trials, and I found out the baseline is always fluctuating and they're changing. So I'm wondering the change of the DFF response due to the mismatch can be separated from the baseline. Yeah, I may be separated. Yeah. And then I used the peri-stimulus time histogram next. And yeah, so my analysis will focus on the block 1 and block 2, including the orientation-specific mismatch within the blocks, the baseline analysis, and a little context-dependent adaptation analysis across the blocks, across B1 to B2. And the level analysis is done in the RI levels and the mouse time, the DMD unit level, because I treat the 2 DMD as the separate unit. Yeah. And then is my first data, but this one is only to show that the standard is different from the oppo response. So this is one example ROI per stimulus time histograms. The plot A, graph A is in block 1 and graph B is in block 2. So at the level of a single ROI, you can see from the black line, which is the standard stimulus, it's relatively stable across the pre-stimulus, stimulus, and post-stimulus windows, whereas the two oppo orientations seems to exhibit quite distinct response patterns across this temporal profile. So my question is not how standard different from oppo, but is there any difference between these two oppo orientations? Like, is it the larger orientation will have a larger response or other explanations? So the first things I did is to compare the ROI-wise response to standard and oppo stimuli. In this graph, it is a example ROI level standard mismatch scaling in block 1 in both DMDs, and each point represents one ROI, and the x-axis is the response to the standard stimulus, which is 0 degrees, and the y-axis is the oppo response, and there are two. The blue represents the 90 degrees oppo, and the red represents the 45 degrees oppo. So, and I then fitted the regression lines for the two oppo orientations, and in the graph A and C, which is in the absolute DFF measures, you can see the slopes are quite close to the unity, while in graph B and D, which is the relative measure, you can see the slopes is quite different and they're less stable. So for graph B, the slopes for the 90 degrees is 0.73, which is slightly larger than the oppo 45 slopes, but it's quite reverse in the DMD2. The slopes for the 90 orientation is 0.68, while for the 45 degrees is 1.44. So this difference, I gather all of the slopes and then summarize them in a spaghetti plot. So,
Alicia?
yeah.
You have a questions from Stefan.
Okay.
Quick question on this graph, trying to understand you're interpreting the slope as being the important thing here, but you don't force the regression to go through 0, 0. Do you interpret the intercept as well? Or because in a lot of the cases, right, when you've got noise and you simply add the slope of regressions has the tendency to drop because it has the tendency to fit an intercept as well. It's just a statistical quirk on how regression works. So is it something which you've looked at to try to force the lines through 0, 0?
I know this is a big limitation of my project, and I focus mainly on the slopes, but not really the intercept, because
Okay.
when I try to fit these dots, and some of these are look uh, quite nice, but when there are like more RIs, they're mainly like clustered in one part, or they may cluster like vertically, and they behave like quite differently. When I also use like the trimming methods, but it doesn't really helps. Yes.
Okay.
So
Thank you.
Okay.
Also, one other clarifying question. Also, I'm a little bit confused. So in here on the A plot, that is 0 versus 90. Do you happen to have a graph in which the same stimulus is an oddball or not? Because sometimes you might look at what is an oddball, or you might simply look at what is the coding of a particular orientation.
Yes, so I think the code for distinguish the oddball and the standard is I'm adapted from the Jerome's code, and it helps me. Um, one measure is the orientation distinguish, and I think another is the um, comparisons. Um, I'm not sorry. I'm
No worries.
yeah.
It's a valid
Yeah.
measure to do. It's just a different type of interpretation for the code. Thanks. Sorry, sir.
Yeah. Okay.
Sarah.
Sorry.
Yeah, just a quick comment. I think what Stefan says makes sense. I think we were just trying to be very pragmatic and just ask, is there a difference in terms of the overall scatter of the data using the slope to say, to some extent the slopes differ. But one thing Alethea ran into is really worries about overfitting, and she tried many different ways, but honestly, I think she never really came to a conclusion that she solved the problem. And regarding the specificity of the responses, she compared the two oddball blocks, but I think we haven't gone to the level of saying how does a synapse that responds strongly to 90 in block 1 behave when it becomes basically, for instance, potentially standard in block 2.
Thanks.
Can I ask you, Alicia, some interpretation of this plot? Because if we focus on panel A, I think this plot shows that the units that respond strongly to a particular oddball also respond strongly to the standard at 0 degree. Is that correct?
Yeah. Yeah.
Okay.
But
So
this is the one. It is one ROIs because
that's
it
right.
is in one mouse in
That's
one
right.
DMD1. Yeah. So it is
When you
the single
plot
ROI. Yeah.
when we plot the DFF without this baseline correction, you see really strong correlation between the two orientations. And so I wonder if a correct interpretation of this is that the cells that have, or the synapses that have strong sort of signal will sort of have a larger DFF, and the synapses that have weak signal will have a smaller DFF, and that translate to all of the stimuli. Is that a correct interpretation, or do you have a different one?
I think it's quite similar. Yeah.
And
Because yeah.
Okay, thank you. I think we're on the same page. I would just let you continue.
Okay. Yeah. Because like this is true because for all of the absolute comparison, like for nearly like all of the mice, their absolute plot comparison, like the single ROI is responding strongly to both standard and oddball. So that's why their line in the absolute plot is always close to unity, and is quite good to visualize. But when I do the baseline subtraction plot, and they remains quite hard to use the linear regression to fit them. Yeah, and it appeared quite is not that stable and quite hard to analyze. Yeah. But as Sarah said before, I never reached a very good strategy to solve this problem. So I focus on the slopes directly. Yeah.
That's okay. I mean, we
Yeah.
can help out. I think there are strategies to correct for the baseline expression level, but I think this analysis is trying to control for.
Yeah. These are the summarized plot, which I gather all of the slopes, the fit slopes, in both absolute and relative measures, and they both in block 1 and also the block 2 in their later stats, in the later slides, they both show no consistent orientation specific output effect across the units. Yeah, using these measures. But I would also be questionable about these measures due to the limitation of linear regression. Yeah, but when I simply using these measures, the slopes didn't change significantly between the output 45 and output 90s. Yeah, and this is the block 2 comparison.
So this is an analysis. This is the relationship between different stimuli is constant in the two blocks. Is that right?
Yeah.
Okay.
And this is across the unit. So before is the example to show how it looks like and how I gather these slopes. Yeah. And because the orientation didn't really explain the mismatch response, so I'm thinking about whether the baseline also contribute to this. And I did the what I call the baseline analysis, and this is also one example in one mice. Yep. So the first thing I did is to use the scatter plots as well to compare the baseline during standards and against the baseline during the mismatch response, which is in graph A to C. And then the second thing I did is to use the Pearson correlation to quantify between the baseline shifts versus the mismatch response shifts. And they're calculated by during one mismatch condition and this baseline minus the baseline during standards. And the last steps I use is the state dependence analysis is to compare between the mismatch response shifts versus the baseline during standard. Yeah. And the across the unit, the population.
Oh. Am I the only one that lost Alicia?
No, I think she froze. Yeah.
Oh. We lost our speaker.
The video froze.
Oh, Alicia,
Sorry, can you hear me now? I think
we lost you for about 30 seconds.
It would be great if
Yeah,
you
sorry,
could go
it's
back
my-
to the if you go back to the previous slide and say what's the takeaway message from your analysis, because we missed, I think, the last part basically for point 3.
Yeah, sorry. My computer like lost for a bit. Yeah. So from point 1 to point 3 is basically the different measures I wanted to analyze for the baseline, and the graph besides is for a single mice, the single DMD1. How is the data presented like? So, but I will mostly explain the cross population across unit analysis here, which I extract the most to relevant analysis results here. So the graph A is the baseline shifts, which is simply calculated by the baseline during mismatch minus the baseline during standard. And to see that the main baseline shifts in different conditions are quite small and is only ranging from the minus 0.001 to 0.009. Yeah. And the graph B is the Pearson correlation graph, which shows that all of the baseline shifts are negatively associated with the mismatch response change. Yeah, I think this is the only strongest with significant difference plots I can get from all of these previous baseline analysis I mentioned, because like from the previous scatter plots or the state dependence analysis, they also didn't show a very significant difference between the conditions. Yes. So
Can I ask you a clarification?
uh-huh.
So panel B, does it indicate that when the baseline decrease, the response increase? Is that right?
Yeah. Mhm.
That's surprising, right?
Yeah, so this is really strange, but I would always say like when the baseline shifts is larger, but the relative mismatch response is tended to be smaller. Yeah, and this is sorry.
What does the baseline is the average activity during 300 milliseconds? Is that right?
Yep.
Okay.
Yep. And I didn't mention that, and it's quite important. I averaged the baseline and the absolute response in each single trials. So I didn't average all of them together across the for one ROIs, if it makes sense.
Sarah?
Yeah.
So just a thought. So, I mean, the mouse doesn't know during the baseline what's gonna show up on the screen, or at least I hope it doesn't, because I think we supposedly randomized it. And so I would be really curious to see the same analysis, but by basically saying, how does a stimulus affect what's coming after the stimulus when the gray screen comes on again? So essentially just shift everything by one time window to basically ask, does an experiencing an oddball have a strong relationship with what the baseline does shortly after? Because that might explain more that there's some big changes, because otherwise I don't really know what to think of this, because the mouse, strictly speaking, shouldn't have an idea of what's to come for all of the stimulus, even omission.
Alex?
Quick technical question. So the baseline that you have here on the y-axis is absolute. What about the responses for the oddballs and the omission? Are they absolute or relative responses?
They're the relative response. Yes.
I fear that then this might be a trivial result. Do you see why?
Yeah, I know, because it is calculated from like the relative is calculated from like absolute to minus the baseline. So I see your point. Yeah, but because when I hypothesize that the relative response is the change of when the stimulus comes, there is a change. So I'm thinking like whether this change can be having some relationship with the baseline. So I don't really think it is a very good measures to show like this is a I would not say like this is the real causal relationship because of this calculation trivial. Yeah.
Well, I think technically speaking, you're having two orthogonal axes, so they should be independent measures. But your x-axis already depends on the y-axis, because for calculating your x-axis, you're using the value on the y-axis. You're subtracting that baseline that you show on the y-axis. So one thing you could do is you could have absolute responses on both sides. So an absolute baseline and an absolute response on the x-axis.
I mean,
Okay.
if
Yeah.
if the response to an orientation depends on the baseline activity in the last 300 milliseconds, that could be interesting because it could say, you know, for instance, that
Mhm.
the synapse is depleted or like not used for a while, and so you have a stronger response after that. But I think Alex is right, but we should be careful with any potential statistical artifacts.
Let me make it more clear. So you're saying I'm taking a response, let's say that's 5 or 7 or 8, and I'm subtracting the baseline, right?
Yep.
Well,
Mhm.
now
Yep.
let's say the baseline is 2. So you're subtracting 2 from each of these numbers, right?
Mhm.
And
Yep.
let's think the baseline is 5. Now you're subtracting 5 from each of these numbers. Well, then what you would expect is that the higher the baseline was, the smaller should be the response that's remaining. Do you see?
Yeah. Yeah. I see
So
this
that's
point.
unfortunately I think that's what you discovered here.
Okay. Yeah. Okay. Sarah.
Yeah, I just would like to add, I understand the comment. I just don't think it's necessarily trivial to think that there are right shifts in the baseline. So basically, she started with doing stimulus evoked activity, and then she realized that there might be really something fundamental at the level of the baseline that's just fluctuating way more, and that's kind of her rationale to do the analysis the way she's done it. But yeah, it's not necessarily just looking at stimulus evoked activity the way you would say. But I agree that the correlation between absolute baseline and absolute evoked response would be a very nice addition.
Well, this one, again, what you do, if the baseline is higher, you're subtracting more. So that's why you get less of a response. It's entirely trivial, unfortunately.
No, but that's the definition of stimulus evoked activity, right? The truth is baseline fluctuates as a function of internal state and other things. And so if you want to study sensory processing, you have to factor in the ongoing activity. And the truth might be that sometimes the ongoing activity is higher. But if you want to decode why an animal, processes sensory input in a certain way, you have to take that into account, and it might reduce the stimulus evoked activity. But I think that's just biology, right? You can't just look at only absolute responses because they might not really, right, reflect what's actually being processed based on the ongoing state.
I agree with all of that, but the specific analysis that you're doing is that your definition of a stimulus evoked response is we have to subtract the baseline, right? And so if you do that, then you will find in every single study, every measure, that the higher the baseline was, the smaller is your response. Because you're subtracting more and more baseline from the response. Now you could say, well, that's what we found is there's nothing compensating. There's nothing compensating for the baseline. So you'd have said, if we wouldn't have found this finding, then if the baseline goes up, the response also goes up. So I think that's fair, that you're showing that by subtracting the baseline, you do reduce responses. So nothing is compensating for an elevated baseline. I think that is fair.
Yeah.
Okay. Yep. Yeah, I think I will take this. So sorry.
But, Alicia, this is a great discussion. I think don't feel like we're questioning your work at all. Like it's really important to have sound statistics agreed upon. Stefan?
Just a quick comment here is that you could easily imagine a little bit on both sides of the arguments that once you raise the baseline, you could have higher activity on top. So to subtract it makes sense. At the same time, that is assuming that you have actually a true knowledge onto what the true baseline is. My fear is that the capacity to estimate the baseline is dependent on noise. And sometimes what you call high baseline is actually not truly a high baseline, but unlucky activity during whatever you measure as the baseline part. So what I would suggest is trying to think of a way to do an estimate like this in a bootstrap, rotate activities, or do an estimate of noise to try to have a dependence like this, because this is a tricky statistical question to ask, as Alex mentioned.
Well, actually, to follow up on that, this is becoming an interesting discussion. So what do you mean by noise? Because what I could imagine is that if you make a global baseline, so let's say for the entire DMD, so then it's not noise in the camera or something, right? So at best, it's something like arousal or something that goes up. If the entire all the synapses go up and down in baseline, then you have like a global baseline, and you can look how that affects the local response. And then it would be hard to argue that's noise. For us as experimenters, that's noise. But obviously, the brain doesn't know it's noise. It only knows all my synapses are more active.
Sure, but can you tell us, please, how you computed the baseline here?
I think it's for Alicia.
Oh, for me, how to? Sorry, I didn't catch that.
How did
I
you
was
compute
taking
the
notes.
baseline?
How did I compute a baseline? Is for each trials, and I averaged the mean response during this pre-stimulus window from the minus 0.343 seconds to 0 seconds. Yeah, and this is for each trial is the trial average measures. Yeah.
So, but in total, how many seconds of fluorescence are averaged in a baseline? Order of magnitude.
I think I averaged all of the.
I understood 300 milliseconds.
So it's just a 300 millisecond in one case, right? So it's not averaged across trials. Right, so that one is a very...
Maybe I can help Olivia to explain. So basically, I think the way she calculated is the way you would normally do it when you would do electrophysiology. So you would basically, for every stimulus presentation, you would use the preceding baseline and then integrate over that baseline window to, in this case, get the average fluorescent value. And that's what she basically did for every single stimulus presentation. And then she averaged that across all the stimulus presentations to have an average baseline or an average evoked response. And so it's basically kind of half of the amount of the recording, the way the experiment's been designed. So I think it's at least half an hour of fluorescence measures, basically.
Okay,
At least.
so it's quite a long time, which is going to be, so that is highly unlikely to be subject to small noise variations. Thanks.
I think it's highly likely to be subject to fluctuations in internal state, right? Because every moment is an instance.
Yeah.
And so in that sense, it's a bit non-traditional for calcium imaging data, but it will be probably the most traditional for electrophysiological data.
Right, thank you.
I think we've discussed. Maybe Alicia, you want to continue, and how many? Do you have a lot more slides, or?
Yeah, I think I can quickly go through. Yeah.
I know you don't have to go quick with it. Just wanted to get a sense.
Yeah, because I think the following slides are quite similar, because next slide is also the same in block 2. Yeah. And the last slides with the data slides, well, it might become more controversial, but I plots the pre-standard orientation in versus the post orientation. Like either they have the same standard roles, or they have the same orientation, to compare like between the block 1 and the block 2, and also use the fitted slopes to regress. Yeah. And then gather these slopes together to summarize in these plots. Yeah. But my interpretation is that this context role for may shape the response, but its heterogeneous is quite hard to tell. But like when I compare, for example, in the same role, in the same optimal role, and within the same orientation in graph D, to compare like the pre-optimal 45 versus the post-optimal 45, I think both the absolute slopes comparison is quite stable, it's quite similar, and the relative slopes doesn't shift much. Yeah, but when I shift, when I look at the graph B, the contextual role change with the same orientation from standard zero to oddball zeros, and its fit slopes is changing dramatically, which it may suggest that the response may be organized by this context change. Yeah, but again, so this has a problem with the linear regression limitation, and it also is not that serious because it only have 3 mouse, lacking the block 2 design in 2 mouse. Yeah, so it's only 6 units here. I will only put this as a suggestive evidence in the analysis. Yeah. My focus is in previous slides. Yeah.
Just a quick comment. There is some overlap here with some analysis that Lucas presented a few months ago. And so I think there might be an opportunity to share notes after the meeting in the forum with him or in emails or so.
Yep. Okay. Yep. And so my final conclusion is that the standard and oddball response, it can be distinguished, but I didn't have enough data to support a reliable difference between the oddball orientations. And so I would say the baseline and contextual role may contribute to the mismatch response, but again, I cannot really quantify or have a strong, significant value to support that. Yeah, I would say these at the dendritic level, the response are heterogeneous and it may not like shaped by the stimulus identity alone, but a broader context. Yeah. And the methodological limitation is also shown here, like the linear fits, or I also pull the ROI analysis together and doesn't really focus on their anatomical locations. Yeah. So I would say maybe like if I can further my analysis, I would first to classify the different ROIs into functional groups, like to classify them into facilitated or depressed or unchanged groups. Yeah, and then do the analysis. Yeah, so that's all.
Thank you.
Okay. Thank you. And thank you for the comments. Yeah. Because this is exactly the obstacles I met when I'm studying, and I'm really like having these questions with Sarah as well. Yeah.
I have
Thank you. Oh, go ahead, Joe.
Just a broader context, and then we should keep some time for Carter. Well, first, this is great. Thank you. Second is, I think for the SLAP2 analysis, we will put together a set of notebooks that help people analyze the data. And so I feel like questions about the baseline and how to measure it properly and use it in analysis might actually be worthy adding in those notebooks. That's one. And the third point I want to make is, so we could collaborate with you to put together these notebooks. And the third point I want to make is the EFIS data have similar blocks. And so you could analyze it in the same way and see if your results hold.
Okay.
That was my comment.
Yep.
Alex?
Thank you.
Yeah, I also wanted to say real quick that I was slow to appreciate the baseline finding. I think there's something interesting there. I agree with Sarah now. And I fail to understand that this is trying to get at whether the animal has an expectation. So I think that looking at the baseline is clever. But also I wanted to say your first finding, I also was slow to understand. This is actually interesting because when you showed it, I thought, hey, look, there should be orientation tuning. So if you just look at oddball responses versus the zero response, you might just rediscover orientation tuning. But the fact that you didn't see a big difference between the two oddballs is actually interesting then, right? Because it suggests that in a way it overrides the orientation tuning. So you might find that there's something that the oddball signal is stronger than the orientation tuning itself. So that sounds like worth going after as well. So, but yeah, the baseline finding, I think that would be really interesting then to compare to the neurophysiology. If the neurophysiology shows something different, that might be really exciting.
Electrophysiology, you meant?
We call it neurophysiology because electrophysiology is what you do on the scalp.
Oh, okay. If you, we all recording neurons in my mind, but I think unless there are more questions, we should proceed so that Carter has time to present his notebook. Alicia, thank you for presenting, and then please share your presentation on the forum if you're fine with that.
Okay. Yeah, sure. I can share it. Yeah.
Okay, Carter, do you want to take it away?
That was a great presentation. Yeah, I'm excited to see more about what we see from our SLAP 2 data. So hey, everybody. I made 2 notebooks, one for our mesoscope and one for our neuropixels data sets. So I just want to show you those notebooks today. We didn't make one for SLAP 2 yet because the NWBs are not yet available, but we're close. Let's see. Can you guys see my screen?
Yes.
Okay. So I put these notebooks in the OpenScope databook, which I have talked about. I talked about a long time ago in a predictive processing meeting, but I'll just summarize it for those who are yet unfamiliar with it. So OpenScope databook, it's a public MIST book online. Basically, you can see if you search OpenScope databook, and basically it's a library of analysis notebooks designed to work with our NWBs. Primarily with the Allen and Sear OpenScopes projects. So it spans subjects from just opening the NWB, just pulling out, visualizing like the type of data, like neuropixels, to like higher order stuff, like using Zebra from the Mathis lab to analyze some stuff, or using GLMs. And then we also have this section here, which has one notebook for each of OpenScope's data sets. So for any of our data sets, these would be useful jumping off points. And I think each of these notebooks has basically 2 purposes, which the first one is just as a companion to the paper to just sort of explain the project, right? That actually comes with code. And then the second purpose is if you want to do analysis yourself, you could repurpose the code and adapt it. So I made 2 notebooks here, one for neuropixels, one for mesoscope. And I will just go over this one here. Oh, another note is, so if you're just like running one of these notebooks, I would recommend you running it locally and then installing the dependencies in whatever your preferred environment tool is, like Conda or something. I have found UV is the most useful environment tool. So there's instructions on the front page on how to set up the environment if you need to do that. But I'd recommend looking at that if you're planning running one of these.
Carter, a quick question. So one of my students is working to develop the quality control for mesoscope data. So should I tell him then to start building on this notebook? Basically, this is how he will access mesoscope data through this notebook.
Yeah, I mean, you could easily adapt it. I mean, actually accessing the data is very easy. It's like 4 or maybe 3 cells here. It's like just these. And so then you have the NWB file and you can do whatever you want to it. So you could easily copy this for that. Yeah. And then like the first half of this notebook after that to like around here is just pulling out the fields of the notebook in like a useful format. So a lot of this would be useful for you and your student.
Thank you.
Can I make a comment, Carter?
Yeah.
I mean, it's possible for Zane that there might be separate notebook that needs to be done to look at specific things. And so, yeah, I completely envision that a notebook on QC or checking your mesoscope data quality could be made and could be added to the data book and linked into the data release paper. And so this way you will get 2 publications instead of only one.
Okay.
Yeah, I definitely think that as a as a group, I mean, just for Slab 2, for Mesoscope, like just creating a corpus of notebooks would be a super valuable resource. So I think we can continue to like create this collection. Okay, so oh yeah, Sarah.
Yeah, just to clarify, so the goal of your notebook is basically to illustrate mainly how to access the different data properties. Do you also have steps planned for analysis, or what's the idea in terms of how these different notebooks are supposed to be structured?
Well, I think each notebook can be structured differently, right? I mean, like a QC one, for instance, you'd have, so this notebook does have analysis. It's just, this is kind of designed to be like a from the ground up explanation of the project. So for someone who's going into it, no context will need, before they can do analysis, right, they'll need sections about what the data are and how to pull it out. But this does have like some basic, like basically trial aligned responses and comparing and contrasting the oddball and the standard blocks here.
Okay.
So I think,
makes sense.
Yeah, I think, yeah, this would be like a good template for creating additional notebooks. But this is more comprehensive than what another one would be. Whereas like if you did a QC one, you could maybe more thoroughly dive into specific pieces of the file. Yeah, so I won't go over everything in these files or in these notebooks, but because if you're interested, you can take a look, but I'll just go over the interesting bits. So we have this introduction. This is pulled directly from the review paper. So it's the same text. And just describing the experiment and the design and the context. We have this, the figure describing our cohorts. And then briefly describing what's like actually in our Mesoscope files and what the stimulus are. So we have our environment set up, opening the NWB file, like I mentioned, with just briefly mentioning what the main fields are here. Metadata, so if you want to see what the genotype is or the mouse age, species, etc. And then it pulls out the imaging planes. It gets the depth for each one, shows you how to find the depth in the calcium indicator. But to actually see the first interesting piece of what's actually in the file is we have for each plane, we can see the max projection image and the segmented ROI masks. So each pair here is one plane. So you can see there's quite a lot in this file.
Is that in the NWB?
Yeah.
Nice.
Yeah.
Have you seen how many ROI we have? It's crazy.
It's a lot. Yeah. Oh, I wanted to point out, yeah, so what I haven't done yet is try this out with other NWB files. So actually, let me point that out. So you could take another NWB path and plug it in here and then rerun this notebook. And so you'll be able to just get, I think since all the notebooks or all the NWB files are formatted exactly the same, this would just run the same and show you for another NWB. And to get that file path, people tend to be confused about this. You would go to Dandy, the Dandy set, and you would click on the NWB here, and you'd click on this little metadata tab, and you could copy and paste this path right here. So that tells you which NW to get, and you can paste it in here and rerun that. But to continue on with what's in the file, so we have the planes, the ROI masks. As a reminder, we already have DFF packaged into the file, so that's already done. And this is just pulling out some sample of DFF traces from Viz L4, so from one of the planes. And you can see this nice looking activity here.
Can I jump in really quick? So we had some debates about not using the standard DFF over F that I guess is used in 2Photon, but use a different. Caspar, I think, said we should do it differently for the snap. So I just forget. I'm old. I forget where that stands at.
So here, I think we have a number of different. I always use the DFF when I'm looking at it, but we do include the raw fluorescence trace and then neuropil and then events. So a couple of different traces. So, but if you wanted to recompute your own trace, you could just take the raw fluorescence from the file. And then normalize.
Didn't he say we should do that? We should do that for every response or something. That we shouldn't do a global DFF over F. Was that I think what we settled on?
Can I jump in, Carter?
Yeah.
This is a really important question, and I think depending on the analysis, you answer it differently. So, in this data, it's a pan-neuronal line, and so all neurons are labeled, and so there are inhibitory and excitatory cells in the mixed. And so the inhibitory cells have a very different firing rate than the excitatory cells. They also have different baseline fluctuations. And so depending if you want to look at fast events or like slow state-like changes in the trace, you might have a different baseline. And so I feel like there's not a one answer for all analysis.
Yeah, and we haven't yet written the final code to package the SLAP2 into NWB. So that's we could have an ongoing discussion about what we can put in there, what we're already computing to. But yeah, we'll need to talk to Casper more. Yeah, so it might not resemble what we have for the mesoscope files. Okay, so we have the DFF here for, this is just for one plane, where you can see the y-axis is the somas and the x-axis is the first 600 seconds. And you see this interesting boundary here. I'll get back to that when we see a full session plot here, but I think this is changing context. So this pulls out the behavior data. We use the pupil area, but they also have corneal reflection tracking and then like a whole eye tracking. So you can see we just pull out the traces for the running speed, the eye tracking, and then this is the mean DFF activity for this plane. But they're not yet temporally aligned. So this just cuts off at the 600th, timestamp or whatever. But it does. These are not yet aligned, so you can't compare. And then this is just a review of the stimulus. So what we have available in these in terms of stim tables, and then it pulls out all of the different stim epochs. And this is a sensory motor mismatch session. So you can see that's this big block here. You have the control blocks, and then ending with the receptive field mapping blocks. And we select, in this notebook, we selected halt oddball times. But it gives an overview of what the oddballs that are available here. We got 68 presentations of halt, and then we selected 952 of the normal presentations. And then we produced this aligned overview of the whole session. So I think this is a great plot. So the thing that really sticks out to me is where you can see the epochs, but you can see at basically every single epoch, like context change, you can see the DFF completely changes. Like you can see this really sharp boundary here, which I think was awesome. I didn't look yet in other mice, so I would be interested to see how this, if this is constant for all mice or if we don't see it in every mouse.
And it's not just the running.
Yeah, right.
Because of the
eye tracking, the DFF, everything changes. Yeah, you can see. Yeah. So it's clearly the network activity is responsive to whatever these different blocks are. Okay, and then so that was all just pulling stuff out of the file and aligning it and stuff. So to actually pull out these windows, so I chose 1 second before and 2 seconds after stimulus onset, and we pull out these DFF windows for each ROI. We can average them across trials, and then we produce this so this is a population mean here for VisL4, and you can see very clearly, right? There's this temporal component, which I don't, because this sensory motor mismatch, it's not trial based, but you still see this sort of cyclical thing. So that's interesting. But then you saw obviously this clear contrast in the oddball, right, which persists for 2 of these cycles, and then sort of returns back to normal. Let's see. Oh yeah, and then for this one, I just got windows between the standard and then the oddball, the halt oddball specifically. I sorted them by the peak response, or excuse me, I sorted by the peak response difference, and then got the difference between the two. So you can see there's this sort of these cohorts of cells that are, these ones are much more responsive, these ones are a lot less responsive, and then it kind of tails off here. Yeah, so I think this is like a good intro starting point for mesoscope. Oh, it's worth noting, I wrote this with Claude partly, so you see M dashes and stuff, don't be suspicious. But I did proofread the whole thing and heavily curated it. So yeah. So this is the mesoscope notebook. Are there any questions about this one? Okay. So I'll just briefly go over what we have in the NeuroPixels notebook in the last few minutes here. The top is all the same. Opening the NWB file here, there's 6 probes, and then there's an electrodes table which describes each electrode along all of those 6 probes. When CCF the CCF histology is available, this field will be filled in with the brain region, but right now it's unknown. And there'll be some additional columns that will be the XYZ coordinates. You can take the units from the units table, and each unit in the units table comes with a quality metric for a bunch. Like there's, you can see the columns here. There's tons of columns that you could use to examine all the units. But I picked a handful of them and showed the distribution of these different quality metrics. So some of them we have like very standard quality metrics that we filter based on that I define here. I think for signal to noise ratio and firing rate, I just chose sort of arbitrary ones based on these distributions to filter units. And then I picked probe C to select units based on and sorted them by depth. Oh yeah, here's a spike raster of the units we selected. And I sorted, yeah, probe C by depth. So this is probe C here because I think I recall it on that probe C targets primary visual cortex. So that's why I picked that one. And then this just gets the behavior in the running, just like we already saw, and we get the stimulus block structure. This session was a sequence mismatch, so that's why this looks different. But the structure is mostly the same, except at the end, we also have the opto tagging block here. And then the oddball I picked was a omission. So I think it's the final trial of the sequence is omitted here. And we produce the same running or the same full session plot here, although this isn't super useful, the population mean firing rate, but you do see some boundaries, some interesting boundaries here. And yeah, we also have LFP data in these files, which is basically by depth along probe. So these are some of the traces from our LFP on probe A. And at the top, you can see this is basically just noise. A better view is this, which is trial-aligned LFP activity. And this boundary here, this really sharp boundary, is the probe exiting the brain. So these handful of channels are outside the brain. But you still do see this, this trial-aligned response here in the LFP, which is interesting, worth looking more into. But then to get to the analysis of this one, I chose 200 milliseconds before the stimulus onset and 1 second afterward and got PSDHs. I didn't show the PSDHs, but I produced them here. And I calculated the spike count distribution in the response window. And you can see they look, they have this sort of Poisson distribution, I guess. And I picked a sample of units. I think this is 50 units, and compared the oddball and standard distributions between them. There's just not enough trials to get really good distributions for oddball, but for standard, you see there's basically two classes of cells. There's this standard, this Gaussian distribution, and then there's this like Poisson, I guess, distribution here. So that was interesting. And let's see. Oh yeah, then yeah, the actually interesting stuff here is I averaged across trials, compare between oddball and standard. And you can see here on the secondary x-axis is the temporal structure of the trials is marked. And so you can see very clear temporal structure in the responsiveness. And these are also sorted by the peak response, I think. So that's why you see it down here. And then I z-scored them. I took a pooled baseline from both, and this is a subsample of the standard trial average activity. I z-scored them. This is not as clear as what we saw in the OFIS, but you do see a couple of things. So firstly, if you recall from above, these were sorted by depth. So this like mid-depth area shows like basically just noise or something unresponsive, mostly. But then among a small number of neurons or a small fraction of them, you can see this very clear differential. I subtracted the oddball from the standard z-score, right? And then you can see there's a number of cells which are just unresponsive here in the oddball. And then there's a handful which seem to be like at the end of the first trial, they seem responsive. All right. And then the last thing is basically the same plot we got here. So the eye tracking, that merits some more looking into. I don't quite know why there's this big differential here, but the population mean firing rate is, you still see this repetition, and you do see this very stark distinction. So, and then it sort of returns. So yeah, I would encourage just pulling the code from these. If you're interested in doing analysis, I think it could be useful. And most importantly, I think if you have people who need an introduction to the project and the data set, I think these notebooks would be really useful resources to point people to. Are there any other questions about these?
Yeah, from Lucas?
Yeah.
Oh, sorry. This was accidental. I just wanted to clap. Very nice.
Oh, okay. Okay.
But maybe just a very quick question. You had this one part with you said they were unaligned, and then you showed another plot where, I think this was mesoscope still.
Oh, yeah. So I do align them. I interpolate basically all of the timestamps to be the same time axis. So
yeah, can you just quickly explain how you do that?
Yeah, I use
necessary
SciPy.
to
I just
line?
use the
Because I
SciPy
thought they
interpol.
share this timestamp.
Sorry, go ahead. Say it again.
Do they share the timestamps, or how do you line? Yeah.
Yeah. So eye tracking and running speed and the DFF are all have their own timestamps when they come out. So these are all actually independent time axes, even though they're displayed aligned. But I interpolate them down here. Let's see. And this is just basically using SciPy interpolate with the nearest. Yeah. So it's this looks kind of complicated, but the actual important line here is, let's see, interpolate 1D, and you just select the nearest interpolation, and so it just produces one common time axis for all.
Okay.
Yeah.
Gotcha. Yeah.
Yeah.
Thanks.
Yeah.
Are there urgent questions? So it's 10:03. I just want to be knowledgeable about time. Are there important questions for Carter to answer? Well, I want to say, Alicia, you're grounded now. You have the notebooks to get started on the EFIS.
Yes. Yeah.
Thank you.
Yes.
Thanks, Carter. This was wonderful. A lot of work, and there's still a lot of work to sort of do the QC and do this analysis across a lot of units. This is just meant to be an introduction to get this work started.
Yeah.
Okay.
Yeah, and I do think we could produce a lot of good additional notebooks that do deeper dives.
I want to sort of discuss next week. We don't have a full agenda yet. I think I'm going to reach out to a couple of you that are working on some of this analysis to see if someone would like to present any update. And then, depending on the answer, I might actually present a potential modeling angle on this data that I've been thinking about in the last few months. So that might be interested about EI balance and things like that. Otherwise, okay. Any other comments? Oh, thanks everyone.
Thanks a lot. Bye bye.